All right, we're live. I want to just thank everybody for joining us today for today's talk from MindCORE at the University of Pennsylvania. My name is Heather Calvert, and I'm the Executive Director for MindCORE, which is the Mind Center for Outreach, Research, and Education at the University of Pennsylvania. MindCORE is an interdisciplinary effort at Penn to understand human intelligence and behavior, and brings together faculty, postdocs, and students with diverse approaches to the study of the mind from Penn's schools of arts and sciences, business, communications, and medicine. Yes, I did read that because I have been skipping that in previous weeks, and so I didn't want to mess it up. So we're live today from my kitchen. My dog is on the floor, and we're joined by MindCORE faculty and a professor in the Department of Psychology, Dr. Allyson Mackey from the Changing Brain Lab. And so I'm going to turn it over. One point of business, if you have questions during Allyson's talk today, during Professor Mackey's talk today, I would like to, I've been reading about how we call women professors, we call them more often by their first name, and that's inappropriate. So, if you can just post them, and you can either post them in the chat, or if you have a question for the end of the talk, it's just a general question. Sometimes the community can answer it, so just go ahead and post it in the chat. You're also welcome to put any kind of emojis in there, like clapping, or I don't know. I see this dancing. That one seems totally inappropriate for what we're doing now, but there it is. And also you're welcome to, you can post a question and ask a question, and then the community can actually upvote your question, and we'll be sure to get to the most voted upon questions at the end. And so that's it for me. I'm going to turn it over to Dr. Mackey.
Thank you so much for having me. Oh, I am echoing.
Oops, sorry.
Okay, sorry, thank you so much. And I'm going to tell you about individual differences in brain development and plasticity, but first I just want to acknowledge how hard it might feel to focus on the mind at a time like this, right? All of our news, all of our world is all about the coronavirus right now, and it might feel like, you know, we should just be working on the virus instead. And when I feel this way, which is often, I remind myself that the people who are working on the virus, who are creating vaccines like Dr. Kizzmekia Corbett, those people were kids once, right? They had to develop the kinds of cognitive skills that they need to be scientists. She had to become curious and persistent to develop strong reasoning skills. And so what we can think about now is how we invest in the next generation of scientists so that we'll have even more people who are ready to solve the world's biggest problems. And so what we work on in the Changing Brain Lab at Penn is the development of the mind, of those kinds of cognitive processes that people need to succeed in science, to succeed in school. And we study how the brain gives rise to those cognitive skills. And we're curious about the kinds of early experiences that shape the developing brain. And a lot of our work so far has been just describing the relationships amongst the environment, the brain and the mind. But where we're ultimately going, and you'll see this towards the end of my talk, is figuring out how we can ultimately change kids' environments so that we can change their brains and change their minds. So we really want to understand these deltas. So to understand how the brain changes, we have to think about brain plasticity. And probably you've been exposed to the idea of plasticity as the process of brain change. So if we think of these two neurons, these two cells, if they fire together, they will wire together. That's Hebbian plasticity. So what we see is if they're repeatedly coactive, we get synapse formation or synapse strengthening. And we also have changes in support cells. So changes in myelination to make the communication faster between these two cells, and changes in glia I just read a paper that came out this week, eat up the myelination that doesn't need to be there anymore, or support the metabolic needs of these cells. But basically we can think of these changes as the process of change. And they've been really well worked out at the cellular level. And we've been able to now start tracking these processes in human brains as well. So we've done work on how adults who practice reasoning as they prepare for the law school admissions test, they show changes in their brain networks that support reasoning, the frontal parietal network. Right? So instead of seeing cells that fire together, wiring together, we see these broad regions in the brain forming stronger connections. We're observing this process of change. Right? So these studies were scanning people before and after learning, but we can also infer that the process of change is occurring by just doing correlational studies. So we see that in kids, if they experience more conversational turns with their parents, so not just overall more words, but really that back and forth rich conversation with parents, then they have more brain activity in language areas here during a language task, and better connectivity amongst language areas in the brain. And so as they experience more language, their brains are wiring up in language circuits. We see something similar with stress. So if kids are repeatedly stressed out, we see changes in sort of emotion regulation circuits of the brain, connections between medial prefrontal cortex and amygdala. And this was work done in my lab by Ann Park, who's a entering 5th year graduate student in psychology. Okay, so this is the kind of plasticity that you've maybe heard the most about. But I also think we have to account for the fact that different people, given the same experiences, their brains will change more or less. So we had some law school admissions test LSAT students whose brains changed quite a lot as they studied, and some who changed almost none at all. Right? So we also need to think about plasticity as the ability to change. And this ability to change drops off pretty substantially as kids grow up. So what we see is that as there's the development of myelination to make the communication between these cells more efficient, that myelination also acts like glue so that there are like fewer changes into the future. We also see the formation of something called perineuronal nets, which you can just think of as a cage surrounding these neurons to prevent future synaptic changes. And development of inhibition, so that when this cell fires, the response in this cell is weaker, and the functions of these two cells become more and more different. And so this is just 3 of many, many processes that happen over the course of development that make our brains less plastic. And that's good, that's what we want. All of you have adult, beautiful brains. You've worked really hard to get your brains to where they are today, and you don't want your experiences tomorrow to just completely unravel all of it. But sometimes you do want to learn big things, right? You want your brains to change quite a lot. And we have the ability to do that because we have neuromodulators like dopamine and like acetylcholine that through motivation and attention, we can sort of reinstill juvenile-like plasticity. Right? So it's really never too late to reawaken brain plasticity. So collectively, we can think that we have these sets of brakes on plasticity, and we have a set of boosts to plasticity. And they change over the course of development, but not in the same way over the whole brain. So this is just to orient you, the side of a brain, the eyeball would be here. This is the back, the top, and the bottom. And it looks like a rainbow because it's colored by different networks that do different things. So in the back, we have our visual areas. In the middle, we have areas that control our body, our motor areas, and our sensory areas. These big orange swaths, those are our frontoparietal network. These are the areas that changed as adults practice reasoning. These parts of the brain do really high-level cognitive skills. They're focused on the external world. Whenever you feel like you're concentrating really, really hard, you're activating your frontoparietal network. In contrast, whenever you're just relaxing and you're thinking about your past or your future or the minds of your friends, then you're engaging your default mode. And it's called default because that's what people do most of the time. Right? Most of the time, we just let our minds wander. And what we see is that developmental processes occur at very different times in these different networks. So we can sort of, as a model, chart plasticity on the y-axis and age on the x-axis, and observe that sensory areas like vision develop first. So they start off with high plasticity drops off over development, and then ends lower in adulthood. Right? These changes happen slightly later in motor networks, and latest still in frontoparietal and default mode networks. Right? And by adulthood, we still think that there's some sort of rank ordering where these frontal parietal networks that do complex cognitive skills retain high levels of plasticity even in adulthood. But this is just a cartoon, right? We could spend an entire career just labeling these axes. What we really want to know is how we can start measuring these things in adults and in humans, and understanding individual differences here. So these might be the prototypical curves, but we might see that some individuals have curves like this, right? They're developing earlier, they're losing plasticity earlier, which might present some challenges for learning. So we want to understand how children's experiences could shift these windows of plasticity. And there's been some work done on thinking about what kinds of experiences might matter for plasticity. So the most important diagonal or sort of axis is probably thinking about positive versus negative experiences. You can also think about how often experiences happen. Repetitive experiences are going to be more likely to drive that process of plasticity, right? Cells that fire together, wire together, whereas rare experiences might do something really different. And just to give you a sense of how you might tile this space, we can think of positive and repetitive experiences as something like having dinner with your family at the same time every night, or always getting to read a book before bed. Experiences that might be repetitive or chronic and negative might be experiences like boredom, or more severe experiences like the adverse childhood experiences, the ACEs experiences. So things like a family member with mental illness or family member who is incarcerated, neglect, abuse, these things that can be really negative and chronic. In contrast, you can imagine experiences that are brief, but still fairly negative, right? A death in the family, a car accident. And there's been much less attention paid to this last quadrant, so experiences that are rare and positive. So experiences like a surprise party or visiting the Grand Canyon. These experiences that trigger surprise and awe, it might be most likely to lead to a dopamine jolt. And so our working model is these kinds of repetitive and negative experiences are going to drive up brakes on plasticity, whereas experiences that are rare and positive are going to boost plasticity. But if we start to understand these experiences better, right, we can start thinking about what kinds of interventions can rescue plasticity, even if some early experiences have driven people to grow up too fast and to have less plasticity. Okay, so just to revisit this, these kinds of experiences tend to cluster together. So kids from lower socioeconomic status backgrounds, so lower family incomes, more likely to experience poverty, are going to be disproportionately at risk for these kinds of negative experiences like ACEs, adverse childhood experiences, and have less access to positive experiences on average. But what's really critical here is there's a lot of variability, right? They're going to be kids from low SES backgrounds who have none of these risk factors. And there's going to be kids from high SES backgrounds that have all of them. Right, so this is really just a first pass to understand these patterns in general. And so what we've seen is that kids from lower income backgrounds show earlier brain development as measured by cortical thinning. So we see this first in young kids between the ages of 4 and 7 in the back of the brain in those early developing visual areas. But then if we look in adolescents, what we see is the differences are much, much broader, sort of over a lot of the brain. Sort of hinting that kids from lower SES backgrounds, on average, their brains are aging or growing up faster. We've seen that in terms of functional development too, when we look at how the networks in cortex become wired up. We see that early in childhood, kids from higher SES neighborhoods now have brains that look younger, but then they have more opportunity and room for growth as they grow up. And so they end up with more efficient networks in adulthood. We see this pattern too in a large literature about SES and stress more generally, speeding up the development of the body. So we see that kids who've experienced early life stress hit puberty earlier. And if you look down into their cells, you see earlier epigenetic signs of aging. So telomeres, the end caps on chromosomes, are shorter in kids from low SES backgrounds. They have more methylation in areas that track with aging. And so this makes us think that if you could tell if a kid's body was growing up too fast, right, that they were hitting puberty earlier, that they had these epigenetic signs of aging, then that might tell you, okay, this kid is at risk for faster brain development and lower brain plasticity. The problem with the measures that we have looking at puberty and methylation is puberty happens too late to really have a good chance at changing things. And epigenetic measures are not very diagnostic on the individual level yet. So we've been thinking about other ways that we can sort of use body maturation as a way to predict brain maturation so that we have more opportunity for intervention. And this is work that Cassidy McDermott, who's just entering her second year in graduate school in the clinical program in psychology, she's been doing this kind of work where she's looking at molar eruption. So when kids get their 6-year molars, which is a very creative and sort of different use of MRI, but this is an MRI of the side of a kid's cheek. You can see their molar coming in here. This molar is not erupted at all. This molar is starting to erupt. This one's almost all the way there, but it's not an occlusion. It's not in line with the other teeth. And at a stage 4, it's fully erupted. Right, so this is something that's happening early in childhood. And we wanted to know how this related to kids' experiences. So what Cassidy found was that kids who've had 2 or more of those adverse childhood experiences have the earliest molar eruption. And so they're starting to get molars even by age 4. That's the purple line. 1 childhood experience is intermediate. Kids with 0 have the youngest looking molars. This is true also for family income. Kids from lower income backgrounds in purple, they have more molars. They have an older looking mouth than kids from higher income backgrounds here in blue. And if you look just at the kids who have all of their molars already, you can see that that's happening around, age 5, age 6 for kids from lower income backgrounds, right? Much earlier. And she's since been able to replicate this in a much larger data set. And what we want to know now is whether this early molar eruption is indeed a risk factor for faster brain development, for learning challenges in school later on. Okay, so bringing that back to this framework, what we see is that lower SES in general is associated with faster aging, right? But again, I want to reiterate that for a lot of kids from low SES backgrounds, this is not true. And so being able to figure out which kids are indeed growing up too fast as early as possible is a major direction for our research program. We next want to know whether there's any impact of lower SES or early life stress on dopamine systems, right? These boosts to plasticity. So does SES impact dopamine system development? And this is work in the lab led by Ann Park. And just to map this out for you, the dopamine forecortex is created by the structure called ventral tegmental area, or VTA, in the midbrain. And it sends projections all over the brain, but most strongly to medial prefrontal cortex. And VTA and dopamine are usually studied in the context of reward, right? So when something is really surprising, really awe-inspiring, very positive, you get this boost of VTA, or of dopamine from VTA. Okay. But it's also important for plasticity. And so what she wanted to know was how the development of connections from the VTA to the rest of the brain differed by early life stress and SES. Okay. So what she's going to do is she's going to look at functional connectivity from the VTA to the rest of the brain, and how SES influences developmental trajectories. And what she finds is really this very specific pattern in medial prefrontal cortex, where kids from higher SES backgrounds show a big, steep increase in connectivity between VTA and MPFC, right, over childhood from age 4 to 10, whereas our kids from lower SES backgrounds are showing a much flatter pattern. And we really don't know exactly why this happens. So there's some work in animal models suggesting that early life stress changes gene expression in the VTA and changes how those cells behave. But you can also think about sort of psychological experiences. If the world in general is not as good as you expect it to be, you might have sort of less use of this system, right? Less occasion for your reward system to be activated. We see a really similar pattern if we try to break down SES and look at stress exposure specifically. So if we look at adverse childhood experiences here, we see a really similar pattern, such that over age, connectivity between VTA and MPFC goes up for kids with no adverse experiences and goes down for kids with 2 or more, slightly. We're seeing another cluster in parietal cortex here too. Right? And this is true even controlling for socioeconomic status. So what it looks like is that lower SES and early life stress are both associated with blunted dopamine system development, with consequences potentially for reward processing, but also potentially for plasticity as well. So what we need to do next is think about how we can use this information to start creating interventions to enhance brain plasticity in kids. So I just want to tell you how we're thinking about doing this. Okay. So first, we can think about how we can reduce biological aging, right? So reduce that process of early molars, early epigenetic aging, early puberty. And there's a good evidence that if we could decrease kids' stress, and less evidence, but I think it's promising to think about increasing their positive experiences, that we might be able to do that. Yeah. And these kinds of interventions actually already exist. So we've been working with a program called Parent Child Plus, and it's an international home visiting organization, and they provide new books and new toys every week. So it's like 92 or 96 visits from age 2 to age 4. And at all of those visits, a member of that family's community comes in and talks with the parent about how to get services, how to avoid stress, how to talk and play with their kids. And they sort of provide this ongoing support to help parents. And so I think it's really interesting to think about how this, like, we definitely know we're getting supplemented cognitive enrichment. There's some evidence to believe that they're decreasing parent stress, how that's going to impact biological aging in the long run. Okay, so we can also think about more directly trying to target the dopamine system. Yeah. And so we can think about trying to enhance curiosity. So we know from many fMRI studies in adults that when people are more curious, you get more VTA activation and more connectivity between VTA, other reward areas, and PFC regions, or enhancing persistence. And I'll come back to that in a minute. So we are working right now with Elizabeth Bonowitz, who is at Harvard Graduate School of Education, thinking about what it would take to enhance young children's curiosity. And she's done really elegant work showing that when kids experience awe, right, which we think is linked to dopamine, they become more curious, they explore more. When they feel empowered to make choices, they explore more. When they feel surprise, they explore more. And now we want to know if kids who practice asking questions, so practice being more curious, you can think of that as, again, trying to leverage that process of plasticity, right? If they practice being more curious, do they become globally more curious? And does that mean that they can then learn better? So again, we're looking to research in adults, suggesting that when adults are curious about new information, I'm sure this connects with many of your experiences as college students, when adults are more curious, they learn better. And so can we actually get kids to be more curious, and does that help them learn more? And so we've created, Sophie Sharp and Greer Bizzle-Hatcher, who's a MindCORE fellow, they've created a science learning program. So based on sort of federal next generation science standards for what kindergartners should be learning in science. So it's based on a book called We Build Our Home, so learning about animals' habitats and their niches. And it's beautiful. The program they've designed is so nice. It has a story. Each lesson has a story, a video, and an activity to teach kids about new sort of elements of animals' niches. And then we can see, okay, if half of kids practice asking questions and the other half sort of get instruction as normal and they're just practicing attention, do the kids who ask more questions learn more? And ultimately, when we can come back in and scan, we can look at how brains change as a result of this. So do we see that when kids have practice with being curious, they get stronger connectivity between VTA and NPFC? And Christina Recto is looking at, she's another MindCORE fellow, looking at our cross-sectional data that we've already collected to see how different measures of brain structure and connectivity relate to curiosity, to give us a sense of what our targets should be for these kinds of interventions. Okay. We are also interested in trying to enhance motivation dopamine processes through enhancing persistence. And this is work led by Julia Leonard, who's a MindCORE postdoctoral fellow, and she'll be starting her own lab at Yale in 2021. So if you're interested in this kind of work and would think about doing a PhD with Dr. Leonard, she's taking students this cycle. What she started with is looking at how parents with persistent kids behave differently. And so we brought in, I think, almost 100 kids or families at this point, and we said, okay, here's a really tricky puzzle. It's from an IQ test, so we know that kids have never played with it before. See how many of these patterns your kid can complete. And then we just coded all of the different parent behaviors that we could think of. We looked at how much parents encouraged their kids. We looked at how much parents gave direct instructions about what to do next, how much parents asked their kids questions, and how much parents actually had to reach in, touch the box, take over for the kids. And of all of the things that we coded, the only one that was really strongly significant was taking over. So parents who take over more rate their kids as less persistent. And this is really nice. This sort of gives us a hint that taking over might be problematic for persistence. But it's just a correlation. So it's possible that parents sort of have insight into their kids and think, okay, my kid's not that persistent, I'm just going to take over for them. And so the next thing we did is figure out whether taking over really can have a causal influence on persistence. And so Julia ran this experiment at the Please Touch Museum. She had these exact same puzzles, and experimenters either took over and touched the box and did the puzzle for the kids, that's the blue condition here, or patiently taught the kids how to complete these puzzles. And what you see as compared to baseline, where kids are just given this puzzle box and they see how long they work on it to try to get it open, kids who had an experimenter take over for them persist much less. And that patient teaching condition didn't really change things as compared to baseline. So it's really a negative effect of taking over for kids, where if you jump in and solve a puzzle for them, they are unlikely to persist on this puzzle box afterwards, even though the puzzles look really, really different. This paper has been really influential for me because I find it really hard to not take over. I think even I was working with Julia on this project and I wanted to make a figure for her just because it's so hard for me to watch people struggle. What I learned is that when you sort of jump in and you take over for a postdoc fellow or for a child, what they're learning is that you think they can't do it themselves. And so through this whole lockdown process, I have 2 young kids. I have to keep this in my mind when they're struggling to tie their shoes or it's really tying shoes that's driving me crazy right now. There are many, many kinds of experiences where it's so tempting to jump in and take over. And I have to remind myself that that can be problematic for persistence. So we can think about how we can actually then bundle that into an intervention for parents more broadly to help boost kids' persistence, boost these dopamine processes, and boost their ability to learn when they get to school. So this whole study was trying to take advantage of learning from individual differences across kids, right? So what is different about those kids who are really persistent versus the kids who are not so persistent? But we also know that kids differ within themselves, right? So we know that some kids will have good days when they're very persistent, and they'll have bad days when they're not especially persistent, right? So a 3-year-old might be really cooperative happily brush their teeth for a long time on one day, and then the very next day not be able to persist at all. And this is a real problem for developmental psychology, because when we bring kids into the lab and we test them once and we scan them once, we really don't know if we're capturing them on a good day or a bad day. So we need to completely reimagine our paradigm to collect more data to sort of get a better estimate of an individual's kid's persistence, but also understand the kinds of factors that move their behavior. So what we did is we asked parents daily questions on factors we thought might shape toddlers' persistence. These are 3-year-olds. So we asked them to report when kids went to sleep, when they woke up, what kind of mood they were in, how stressed out the parent was feeling. And we had them send us videos of nightly toothbrushing over 2 weeks. So from those videos, we can code how long a kid is brushing their teeth for, and we can also code the parent-child interactions. A lot of this data, about 3/4 of it, was collected during COVID. This is a really nice study that we can do completely remotely. And so I'll tell you a little bit about how we think COVID is impacting these findings. Oh, so sorry. But first, I want to see if I can do a video. This is going to take me a second. Okay. Okay, so you can see that this is a pretty good night. This kid is brushing his teeth. The parent is encouraging the kid a lot. But we're going to do that same video every night for 2 weeks, and you start to see a lot of variability. And you can ask which of these factors are related to better brushing nights, more persistence. And I'm sorry if this is small, but what we see is a significant but small effect of sleep. So when kids within child, when they sleep better the night before, they brush their teeth longer the next day. They are more persistent the next day. You have some kids who have stronger effects than others. We're very interested in why some kids are more sensitive to sleep than others. We see a similar effect size for mood. So when kids are in a better mood, they're more likely to brush for longer. The kids who are sensitive to sleep are not the same kids who are sensitive to mood. So it's not just that when kids sleep better, they're in a better mood, so they brush longer. There seems to be another path from sleep. We don't see an effect of parent stress on brushing time. And what you see is there's a completely flat general trend, but you see this fanning out so that you do have some kids who are very sensitive to parent stress and some kids who are sensitive in the other direction. We're trying to figure out why that might be. And last, the very best predictor is how much parents are encouraging their kids. Right? So that's a little bit tricky because it could be that when kids are doing an awesome job, their parents then congratulate them for that. It could also be that parents who are encouraging their kids more, talking with their kids more, are sort of pushing their kids to brush longer. These are really, really strong effects. And so you might wonder what COVID is doing to all of this. And what's surprising is that there's really not a big effect. So within child, the proximal factors that impact their persistence, how they're brushing, are their sleep, their mood, and their parent encouragement. Sort of when we collected this data a year ago or now, no big effect of COVID, even though parent stress is so much higher. So what it looks like to us is that parents are internalizing all of the stress from global events, but locally when they interact with their kids, at least during this sort of very routine time, they're not letting that stress show to their kids. They're protecting their kids from what's going on, and the kids are mostly doing pretty okay, which I found fairly encouraging. So the next thing we want to know to figure out whether these sort of within kid variations are useful for intervention is understanding whether parents already knew this. Did they already know that sleep, for example, was really important for their kids' persistence? And the answer on this is a pretty resounding no. I'm going to move this so you can see it. So we asked parents to, for each of these factors, say, yes, I think this impacts my kids' toothbrushing, or no. So for sleep, mood, encouragement, and parent stress. 82% of parents said they didn't think that sleep mattered. Only 18% said that they thought it did. And then if you look within the kids whose parents thought sleep mattered, that correlation, that sensitivity to sleep is no higher than for parents who thought sleep didn't matter. If we look at mood, almost all of the parents thought that mood mattered, but there's no difference at all in the strength or the sensitivity of the kids whose parents did say mood mattered versus those who said mood didn't matter. And we see this pattern over and over and over again, which hints at the idea that if we created interventions to, say, either improve kids' sleep quality or improve encouragement from parents, that we might really see a change in how kids persist. And then we can sort of do a 2-by-2 design to say, if we intervene on sleep for the kids for whom sleep really matters, do we see a different effect than if we just sort of non-specifically target interventions, if we don't individualize those interventions, which is a really new area, thinking about how to sort of best improve an individual's cognition. Okay, so let me just sum up so we have plenty of time for questions. I think we have lots of good evidence now that children's experiences impact their brain development. I have focused on psychosocial experiences because I have a better sense of how to change those kinds of experiences, but I do want to mention that there are other physical experiences that matter a whole lot. So kids from lower SES backgrounds are going to be more exposed to air pollution and lead and neighborhood gun violence. The list is really quite long, less exposure to green spaces. And so I think we probably need to broaden the field of who needs to be involved in these kinds of decisions of developing interventions to help kids learn. What we see is that some kids from lower SES backgrounds are growing up faster and showing the blunted dopamine system maturation. And we think that those factors will have potential consequences for plasticity. We don't know that yet. It's a direction we're going in the lab. But again, lots of kids who grow up in lower SES backgrounds don't show these factors. They don't grow up faster. They have typical dopamine system development. And so the better that we can identify those kids who are most at risk, the better we can target interventions. I think that probably our most promising avenue is to look at interventions that can reduce stress and increase positive experiences early on when the brain is the most plastic. And then I think we might see benefits that really last for plasticity and learning. But certainly, we don't need to have all interventions start at age 2, right? There are plenty of kids who are in college, right, who still could really benefit from different ways of teaching to enhance brain plasticity. So that's where we turn to sort of trying to translate this animal model literature on attention and motivation, acetylcholine, which I didn't talk as much about, but dopamine and motivation. How can we help people become more curious and more persistent? We've really focused on young kids. That's sort of the area of focus of the lab, where we're trying to boost curiosity and boost persistence by learning from correlations across kids, but also from learning from sort of within subject fluctuations. So to really develop the very best possible interventions to enhance kids' plasticity and learning. So let me just conclude by thanking the lab. I tried to highlight the individuals who are working on this. So if you have questions or you want to get involved, please reach out to them or to me. And I look forward to hearing your questions.
Thank you so much. Super fascinating. And it's always really, I always love this line of work where you actually also talk about the applications. It's not just pure research. It's also like, how are you going to enact some change? So we do have some questions. There's a basic question that showed up early, but I think it's worth asking early so that we have the answer in mind, is how do you measure plasticity?
Such an interesting question, and really the trick, right? So what we can try to do is use imaging methodologies that are sensitive to factors like myelination, start working towards imaging perineuronal nets, which will require ex vivo imaging. So Dylan Tisdall, who's a collaborator of mine, has been doing ex vivo imaging. So after somebody dies, you can scan their brain for a very, very, very long time and start getting better sequences that are sensitive to these cellular processes. The other thing that we can do is just measure how people learn. So Austin Borashak in my lab, who's a clinical graduate student going into his 3rd year, has done some work with adults saying, okay, I'm going to push your brain to learn working memory for an hour, right? How much will your brain reorganize as you learn? So we're sort of pushing that direction as well. And then within kids, I think we have to try to use their learning as a measure of plasticity, right? So as they learn new science information, how much do their brains change? How much do they learn?
If anybody has any other questions, go ahead and post them in either in the chat or the question, and I'll move on to one of our other questions here. This is super interesting. Has there been recent research at Penn or perhaps elsewhere to identify the relationship between maturity, plasticity in children from different racial groups and how SES mediates this relationship?
So I think that's a really interesting question, and I think that there hasn't been that much work yet. What I will say is that I think that being from a racial minority is extremely stressful. So experiencing racism as a parent or as a kid, I mean, you've seen just heartbreaking stories of 6-year-olds experiencing racism. That is almost certainly going to impact these stress factors that might drive in some kids early acceleration in their development. I think these other factors that sort of drive immunological processes like lead exposure, like air pollution, we know that kids of color are disproportionately exposed to these things. There's environmental racism from this history of redlining and sort of deliberately creating structures to basically ensure that kids of color are facing a lot of these environmental hardships. So there's not enough research on it, but I wouldn't be surprised if we do see that kids from certainly Black kids start showing some of these accelerated development trajectories.
It's easy to imagine, even just with the lead crisis in the public schools in Philadelphia, and also I guess I've seen some of the asthma data as well.
Yeah, I mean, health disparities by race. Sorry. Health disparities by race are huge, and they emerge early. And certainly in mental health, they're huge. And so I think trying to figure out in kids, like if they're on this trajectory to grow up too fast, that will help us with better interventions.
Here's a sort of basic question about, not basic, but a question about just the specific studies that you were talking about. Did you exclude children of parents who are diagnosed with mood disorders from your studies?
So we don't exclude based on parent mental health. And I think if you did, you might end up getting a pretty biased sample because depression and anxiety are so much more common in families from low-income backgrounds. Right? It's incredibly stressful to not have enough money, and that can mount and lead to anxiety and depression. So we do ask about it. So we do have that information. It is actually one of the adverse childhood experiences to have a parent with a mental health disorder. It's a really non-specific question, but that certainly will have impacts on the kinds of care that kids are getting. So we haven't looked at that data in a lot of detail, but certainly having a parent with mental illness is going to have an impact.
We have another question that's, is there a relationship between enhancing persistence and the notion of grit in early development? For example, is it valid to view grit as persistence applied to a goal?
Mm-hmm. So I work with Angela Duckworth quite a lot, and she tries to really make a point that grit is both persistence and passion for that long-term goal. So it has a flavor of that curiosity too. So really wanting to know something is lined up with that passion piece of grit. So I think we're absolutely interested in sort of the early phenomena that support grit later on. Kids tend to have, in general, broader interests, broader curiosities. They don't have as specific goals yet. And I don't actually think that we know if early persistence is predictive of grit in adolescence, but that's a really interesting idea.
Thinking, we don't actually have more questions from the audience, but I'm curious about how, I'd like to ask you about where you're going next with your work, but I'm also curious about how being at Penn and living in Philadelphia influenced your decision to examine the SES group more, given that it's the largest poorest city in North America.
Yeah, so I've been interested in income and education since I attended like urban poor schools as a child. And so that sort of social conscience interest is way preceded coming to Penn. And I've been working on socioeconomic status my entire career. Certainly when I was thinking about which faculty job to take, looking at collaborators at Penn like Martha Farah, who really inspired me to see that SES could be tackled from a neuroscience and psychology perspective, being near her was a huge draw. I think seeing Philadelphia as a place, it has really high poverty, but it also has incredible communal engagement. There are so many organizations who really care, who really work hard to do our best for our kids, and sort of I draw a lot of inspiration from that as well. Certainly, if we look at the kids who come in just through our SEPTA ads and social media ads, the demographics are skewed much lower than any of the places I've worked before. So parent education, we have a lot of parents who haven't completed high school. That was not so much the case in Boston. And so it creates challenges for comparing individual difference research in Philadelphia versus in Boston. But I think it's good. It's pushing us towards understanding humans across contexts, right? Because we really want to understand sort of how the human mind and brain are sensitive to these things, not just within Philadelphia, not just within Boston, but much more broadly. So it's really driven me to take those differences in sampling a lot more seriously.
And then what's next? Where are you thinking about pivoting next? Where do you?
So, I mean, I spent a lot of time on future directions in this talk, which I might not do for another audience, but I really wanted to see like what students are excited about, what they might want to be engaged with in the future. And so we have this partnership with Parent Child Plus. The initial plan was to scan all of these kids at age 2 and do a randomized controlled trial, and then scan them again at age 4, watch them go into school. What I think we might do for this fall is just start following them by these remote measures, survey measures, and then scan them after they've had this experience with Parent Child Plus. And so we have to be flexible with COVID to figure out like what data we can still get. We were in the midst of a really large grant to get data on sort of more extreme environmental exposures. So to recruit from Kensington and kids who had parents with opioid use disorders, we aren't able to scan in that way this fall, but I think we're still interested in expanding these remote measures to more diverse populations. We are getting very excited about this intervention piece too. Right, so can we develop really good text message-based interventions to help parents support their kids' sleep and look at impacts on persistence, look at impacts on their curiosity and their learning. So really, like I've been so impressed by people in my lab and how creative they've been able to be in this context. There's a huge amount of data on environmental exposures and brain development from the ABCD study that anybody can get access to. Okay, so we're starting to work with undergraduates in the lab to develop hypotheses, write pre-registrations, and really have undergrads get hands-on experience with huge data. It's 10,000 kids. And so there's huge opportunities to really get a better sense on correlations between environment and brain. And they'll start releasing longitudinal data so we can look at how brains are changing over time as kids go into adolescence.
Fascinating. I can't wait to see what happens next. So I think we don't actually have any more questions.
Great.
So I can release you.
All right.
Thank you so much for joining us today.
I'm going to put my email in the chat. Please reach out if you're interested in any of this and you want to talk more, get involved. And thank you so much to MindCORE and to Heather for organizing. It's just endlessly impressed by the organization able to do.
Thank you. We appreciate it. We've got a great team. So I will let you go. We'll stay live a little bit longer just so people can put their thanks in there. And then as with all of our Wednesday seminars, today's session was actually filmed and will be available at the same link where we attended today. We had so you're welcome to come back and watch it after the fact or share it with somebody. You can just send them the link. And the other thing I wanted to let you know is that next week we have a special event on Tuesday, which is around, it's about scientific posters, and we'll have somebody from the biomedical library. And actually, Julia Leonard will also be joining us to talk about the role of science posters and sort of the share in sharing your research with the wider community. It's kind of a first step for early researchers. And then the biomedical library will give us tips on sort of how to design a good poster. And then on Wednesday, for Wednesday's faculty seminar, we'll be joined by Professor Lila Unger from the Computer Information Science Department here at Penn. And he's been in the news recently, and I'm going to forget about what it was, but it was super fascinating. However, I've been reading a lot of news.
I read something about contact tracing by app and how successful or unsuccessful apps might be. I think he does many, many fascinating things. So that was just one article that I read recently.
That was it. Okay, thank you again. And thanks to everybody for joining us. Have a good rest of your Wednesday. It has stopped raining here in Philadelphia, and we'll see you next week.
Thank you. Bye. Bye.